Hello again. Welcome to week 3, our final video on nucleic acids, and then the very last few slides of this presentation. So the fourth group of the molecule of life is actually, could argue, is the most important because we're going to be talking about nucleic acids. And what we can see here, you're going to have several lectures or a whole week that we're going to be focusing on this pathway, so you don't need to know it now, but just to be introduced to. It's called the central dogma, where this language of nucleotides, or the information in the DNA, gets translated to physically dictate what that sequence of amino acids is that makes up a protein. So it's the sequence of nucleotide that actually determines that sequence of amino acids and makes up each of those proteins that are all the doers in the cell. So getting specifically into nucleic acids. Nucleic acids, like proteins and carbohydrates, have a single monomer, and that is the nucleotide. So nucleic acids are made up of nucleotides. You now will be expected to recognize what a monomer of nucleic acid looks like. It's got 3 key parts. It's got a phosphate group, which again, of course, you recognize with those negative charges due to those oxygens. It also is going to have a 5-carbon sugar. Now, there's 2 different types of nucleotides we're going to see, and there's 2 different types of sugar. One is ribose, the other is deoxyribose. As you can see here, that difference between deoxyribose and ribose is just the deoxy, meaning there is not an oxygen, there is not a hydroxyl group in deoxyribose. Then finally, the third part of a nucleotide is a nitrogen-containing base. Sometimes it's referred to as nitrogenous, just meaning it contains a nitrogen. There are 4 different bases for each type of nucleotide, with a total of 5 bases. Guanine, adenine, cytosine are common in both what we'll find out RNA and DNA. And then uracil looks kind of a lot like thymine because essentially they're almost the same except for this methyl group. And we're going to see that uracil pairs with the ribose, and thymine is going to be only with the deoxyribose. So that is the monomer. Here's a same exact concept, different graphic. Again, love to give you different pictures for the one that sort of clicks with you. And what we're going to see is they come together to form that polymer. So single monomer, single nucleotide, polymer. They get linked together, and they get linked together in a very specific structure. It's got that sugar-phosphate backbone, so the sugar and the phosphates alternate with those nitrogenous bases in the middle. And those nitrogenous bases are going to pair up. We're going to spend many more lectures talking about genetics and DNA and how these pairing up and the history of that. So for now, you need to be able to recognize these structures. You need to be able to recognize that polymer form and that single monomer form, and be able to recognize a monomer is a single phosphate group, that single sugar, and its base. So one of these is going to be a nucleotide. And these bases are going to form hydrogen bonds. You can see that again, those dotted lines are a hydrogen bonds. The straight line is a covalent bond, dotted line, hydrogen bond. So to summarize, we got deoxyribonucleic acid. That's the major hereditary material. It's got 2 nucleotide chains, so that's that famous double helix that you hopefully recognize, whereas RNA is a little bit different, same structure, but it's only a single strand. And again, its sugar is ribose and its base is uracil rather than DNA, which has thymine and that deoxyribose. One of the key things you need to be able to recognize, or one of the key nucleotides that plays a big part, is adenosine triphosphate. So we are going to spend an obscene amount of time, not really, we're going to spend 2 weeks talking about aerobic respiration and photosynthesis, talking about how our cells make and utilize energy. And energy we can think of as lots of different ways, but this is the major way. It's like the currency of our cells. When you need a little boost, ATP. And what ATP is it's simply a ribose sugar with a phosphate group that's got connected multiple, in fact, 3 phosphates groups, because that's what that triphosphate. So we're going to see later on in this class how we go from ADP, which is diphosphate, so 2 phosphate groups, to ATP with 3 phosphate groups. You don't need to know anything about what that energy is, how it's made, but you do need to physically recognize that this is ATP by this structure with that A meaning adenosine, meaning that nitrogenous base. Again, you can see here, there's different types of nitrogenous bases. There's guanine and adenine with those 2 rings versus cytosine, uracil, and thiamine that have single rings, and they're going to form those different hydrogen bonding structures. So those are going to line up and pair up. DNA, as I mentioned, the sequence is really important, and we're going to talk a lot about how that sequence dictates proteins and what happens when you change those proteins later on in the class. So we've got several summary slides here that I just wanted to draw your attention to. Really, it's these ones that sort of, if you're going to be making those review sheets, your review sheet should include all of this information for carbohydrates. Your review sheet for lipids needs to include the different types of lipids that we talked about. Examples, be able to recognize them, know their functions, be able to go from function to component or from an example to its function. Equally for proteins and then for those nucleic acids. So I have some summaries or some you should knows. List and describe the 4 major classes of molecules. Well, that's these 4 that I just mentioned. I think the next question is between monosaccharide, disaccharide, poly. This is, again, root word. You don't have to memorize things. Monosaccharide, that's single glucose. Disaccharide, 2 monosaccharides linked together like sucrose or maltose. Polysaccharides, lots of glucoses linked together to form those 4 different structures we talked about: chitin, cellulose, glycogen, and starch. Saturated and unsaturated fats is all about the hydrogen bonds in those fatty acid tails. If they're full of hydrogens, no double bonds, that's going to be saturated. Unsaturated contains a double bond, contains a clinker, assuming it's a cis or naturally forming unsaturated fat. So I also have this picture. Now, this is sort of your final summary. Can you recognize what all these molecules are? Do you know what each of these is? So I'm going to very quickly go through these, but you need to be doing your practicing to find out. Here we have a bunch of glucose molecules that aren't particularly branched. So this we see as starch. We see a beautiful steroid right here. Here we see that double helix with those bases, those nitrogenous bases paired up. So that's going to be DNA. Here we also see what kind of looks like a helix, and it should because it's an alpha helix, which would be the secondary structure of a protein. Here we see a hydrocarbon. I could ask you if you find a molecule that wouldn't have a charge. Here we're going to have, due to all those non-polar covalent bonds. We have a beautiful amino acid right here with that carboxyl group and amine group, central carbon, that variable group. You can think of R like an X in algebra. Moving right along to number 7. We've got all those glucoses linked in chains, but they've got these hydrogen bondings forming between the hydroxyl groups. That is what we call fiber or cellulose, making up the cell wall. Over here, it did get cut off just a smidge right there, but here we've got a triglyceride. We've got the glycerol backbone, and we've got those 3 fatty acid tails, where we can see this is a saturated, unsaturated, because it's missing those hydrogens. Here we have 2 glucose molecules linked together to make a disaccharide. Here, down here, number 9, again, a whole bunch of glucose molecules, so we know it's a carbohydrate, but we have to think, how is it structured? Is it branched? Is it not branched? Does it have some additional functional groups? Nope. So this is glycogen. You see all the branching of those glucose molecules. Down here for number 12, we see a 5-carbon sugar. We see we just see a C, but it's a nitrogenous base and a phosphate group. That's because it's making up a nucleic acid. 13, we have a teeny tiny phospholipid. Number 14 This is that final, the 4 types of the saccharide, that's chitin. We also have down here just a glycerol. So glycerol and a fatty acid are going to come together to make that triglyceride or that phospholipid once you add that phosphate group. And we can see number 16. Number 16 is a clear picture of glucose. So we can see how this is actually the molecule that's summarized here and here and here and here. Trying to see if there's any place else. I think that's it. And so those are all the macromolecules. I expect you now to be able to recognize the shape of macromolecules and tell me their function. Or if I tell you something's function, to be able to give me what structure. I won't ask you to physically draw a structure out of nowhere, but you are online, so we're going to be working with the software we have. I think that is all. Here's a picture again, and good luck.